of. Let me see. Yeah. All right. So go ahead. We see your slides.
Can you hear me?
Yeah, I can hear you. I can hear you fine. And I see your slides.
Cool. Yeah, my Zoom isn't working very well, so it's actually currently frozen. So I'm going to proceed and ignore it if that's possible. So I apologize, everybody, if I end up having to disappear and reboot, because this happens sometimes with the newest evolution of Windows. My fellowship here, and I'm currently an acting instructor at UW and at Seattle Children's. I undertook this very large, substantial topic per Len, and I have never studied evolution of the immune system, so this will be just as new to me as to you. I've tried to make it very approachable and less technical. Over the last few years, I want to say since 2016, 2010, there hasn't been as much data that's come out of this particular field, but it's all fascinating all the same. So I hope you enjoy the ride with me today. Let's see if I can advance. Okay, I have no disclosures. So the phylogeny of the immune system. So it's incredibly complex, as there's major branches. We know most about systems that are greater than, say, like 450 to 500 million years prior to today. Most importantly, as you may have predicted already, most of these entities, the proteins and pattern recognition receptors and whatnot had actually developed first. Then much later, very, very recently, with mammalia basically, and some reptiles and birds, was the development of more complex organ systems such as the spleen, thymus, MHC, T-cell receptors, antibodies, and other components of the adaptive immune system. So then I split this talk into two major sections, the innate immunity and the adaptive immunity. I feel like that would be the best way to approach understanding how things have evolved over time. Like the last slide indicated, things kind of have progressed through from a protein to a cell-mediated adaptive immune response. Most so-called earlier evolution animals, such as fish and amphibians, it starts with physical chemical barriers, and then later on, do they develop humoral innate immunity, which includes everything from that we know about, from lysozymes, antimicrobial peptides, all the way to cytokines and natural antibodies. And then there's the cell-mediated innate immunity, which was actually really interesting to learn about, because there were cell types that I wasn't actually as familiar with. The phagocytic B cells is one of them. They're also known as the B1 cells. There's actually a lot of literature that's debating the existence of these. So I find it interesting that in the evolutionary scheme of things, people are pretty convinced that they exist. We do know that we are pretty sure that they exist in mouse, but we're not entirely sure, especially that they exist in humans. And I'll be talking about these cells a little bit more later on. And then there's also mucosa.
Did we lose her entirely, video and audio?
Yeah, it certainly looks that way.
Okay.
She said she was going to try and log back in.
Okay. While we're waiting for her to log in, I went to the meeting only for 2 of the business or scientific days, Friday and Saturday, but I found the first plenary session, which was about COVID, as you might imagine, and really very, very well done studies on the human immune response and failure of the human immune response, and reasons, especially for older populations having a high risk. Fascinating. So once the whole thing's posted online, that would be Friday's plenary session, the one that would have started at 8:15. That was really a worthwhile session. I'm sure there were others. Other people may want to chime in who stayed later. I didn't go to Sunday or Monday sessions. But anybody else, while we're waiting for our speaker to give an update or their experience at the meeting, open your microphone and chime in.
This is Vinod here. So I apologize earlier for the microphone being unmuted. So I know yesterday, I think the focus was on the plenary was the microbiome. I think Hugh Sampson and a couple of us talked about, the C-section, vaginal delivery, and, at least he's doing a study of at least trying to swab the C-section born children with vaginal swabbing to see if, kind of restoring the biome could kind of help minimize risk of infection in those population. But I think he's limited by the number of recruitment because of the risk of other infections. So it has a lot of exclusion criteria, but, microbiome was kind of the focus yesterday in food allergy.
Food allergy. So in other words, transferring the microbiome, reducing the risk in a fetus or the newborn of food allergy developing.
Right, atopy in general, and bifidobacterium being kind of the major bacteria. I think there's another study that talked about the, speakers spoke about that.
Other comments about life in general, which isn't too promising right now with a war in the world, is unbelievable. I do not see our speaker. And there's nothing particular. Go ahead.
This is Vinod here again. So I went to one of the morning sessions with Dr. Platts Mills yesterday, and I think he was bringing up an interesting thought about at least in maybe the population that's vulnerable to alpha-gal, in report of association between IgE, again, alpha-gal kind of antibodies with higher risk of, say, cardiac events. That maybe a group in Australia has kind of reported, difference between those who have been had porcine or beef valve replacement versus mechanical valve replacement in terms of their risks. As also, in his select population of, 10 or 15% of so-called IBS, stomach only issues, potentially having, alpha-gal as a problem related to that. But again, not that the IBS population as a whole, but the population in southeastern United States who probably also have, IgE, the alpha-gal potentially could be an issue and could benefit from, their dietary changes.
There was on the exhibit floor, there was a company advertising its alpha-gal deficient pig. I didn't look at it very much, but it's gotten to be a commercial benefit.
So I think they kind of talked about that in that meeting, yesterday. So they have the glycosylation knockoff, whatever pig. And I think there's been two. One, there's been a, person with a kidney transplant from such a pig, and also as well as a heart transplant who has survived 6 weeks. So far, at least there are two transplants from such a pig that's happened recently, is what they talked about. And most, pig kidney transplants don't last. So in this instance, has lasted, I'd say, I think it's been very new, maybe just a couple weeks. So.
Is anybody in communication with Ali? These kind of difficulties remind me as mask mandates get rolled back and the count of new infections really is dropping dramatically. If relatively soon we might have these meetings again at South Lake Union for those who want to come, and then broadcast them. I mean, it's not that we didn't have technical difficulties when we did it that way, but at least there was some collegiality of people getting together. I'd have to get approval, I'm sure, from the University of Washington to go back and use the room at South Lake Union. I don't know, Drew, do you have any comments on that? Who we have to ask?
I haven't heard any guidelines yet. Francis usually knows, but I haven't heard anything new.
I welcome open conversation here because I can't do anything to get our speaker back. Anybody with a case that they want to discuss with a broad group of colleagues? Anybody want else to discuss the meeting?
I just talked to Ali. She's logging back in.
Okay. There was a pretty full exhibit floor of pharma companies at the meeting. If you want to listen to recorded presentations at the meeting, what you need to do is save your registration number. You use the registration number as both your name and your password when you sign in to listen to sessions. The other thing that was interesting, a moderate number, maybe 20% of the speakers didn't come in person, but they pre-recorded their presentations and then had an open microphone for questions. And actually, they pulled that off pretty well. The audio and visual worked at every session that I went to, and then the speaker was open for questions at the end. Hopefully by next year's meeting, these workarounds won't be necessary anymore.
This is Vinod here. I just wanted to say the COVID-related plenary was on Saturday, not on Friday. That's the
That's
presentation
right. Thank you. Yeah, it's Saturday morning. You look at the program. One thing, maybe you knew this already, we've had a speaker from Allakos the last couple of years, and I think it's on the schedule for later in this year. Amal has usually been the speaker, and they've been the company working on a cyclic 8 antibody, with cyclic 8 being a unique marker on eosinophils and mast cells, studying it in eosinophilic GI diseases, particularly those below the esophagus, although they've studied it in EOE as well. And I think through their phase 2 studies, everything looked very promising that this was going to be a very effective molecule. Recent results from their phase 3 study, from what I understand, and this was discussed in one of the sessions, it depleted eosinophils as expected, but there wasn't any clinical improvement, which obviously was a disappointing outcome. So they're looking at the data again to see if there might be an explanation for that. There was another entire session on eosinophilic GI diseases below the esophagus, so more focus on that area. I mean, and one of the comments is the one that I just made, the disappointing clinical data with this molecule. Any of our fellows have comments on your first meeting? Those who went?
Hello, this is Karen, second year fellow. I thought it was a really great meeting to go to. I know we were not able to go last year. It was all virtual last year. I really think you can't replace anything in comparison to an in-person meeting. I met a lot of people and probably made some, networking done that we just could not do last year, and went to several lectures, which and you meet people there. You just bump into people and get to know them. I think at least from a networking standpoint, it was really great. Yeah.
Yeah, I mean, definitely meeting people, being introduced to people who are prominent in the field is pretty much essential for developing your career. So people who never got to go to a meeting during their fellowship, honestly, were cheated. I see Ali's slides coming back up. Is this encouraging? I see her name has popped up now as somebody joining us.
Okay, there we go. Yay, there's a camera now. Sorry, guys.
All right, get on with it.
Where was I? Where did I disappear?
You were on the next slide, I think.
Oh my gosh. This
That's
one?
all. Well, yeah, you had gotten to that slide.
Had I gotten to this one yet?
No.
Okay.
That's where you need to start.
All right, cool. Give me 1 second to navigate. All right. So I've already I was gone for quite a while before I realized that I was muted. So hopefully I remember to include all details the second time around. Please let me know if there's any issues. The chat function should be working now too, but please interrupt me if you have questions. So PRRs, we all know about PAMPs and DAMPs. We don't really talk as much about MAMPs, which are microbe-associated molecular patterns. So DAMPs actually don't arise until much later into, I tried to find some literature about it and there really wasn't any. So my presumption is that it really doesn't start to occur until not just vertebrates, but mammals. Not like receptors, or NLRs detect intracellular bacterial elements, but are not present in Drosophila as well as C. elegans. What is most interesting about this and other PRRs such as RIG-I-like receptors, as well as other PRRs that we talk about all the time with COVID, these actually are not really mentioned in literature. I specifically tried to go find information about these, and they basically focus on everything else, and they don't really talk about those specific kinds of receptors, which makes me think that these also did not develop until much, much later in phylogeny. There we go. So TLRs, like we all know, they sense extracellular bacteria, phagocytosis bacteria, viruses, and fungi. TLRs are classified according to the number of cysteine clusters in ectodomains. So they can either have one or they can have multiple. Ancestors to arthropods have only one single cysteine cluster TLRs, or SCC TLRs, at the C terminal end of the leucine-rich repeat. And then proteosomes such as Drosophila and C. elegans, as well as more ancient phyla, have multiple cysteine cluster TLRs, or MCC TLRs, that contain two LRRCTs on the C terminal end, or they could be on the N terminal end. This particular figure demonstrates prototypical TLRs, which I'll define on the next slide, and compares what an MCC TLR and an SCC TLR basically look like. And then I'll get into more details about what actual TIRs, which are toll-like interleukin-1 receptors, and we'll talk more about what LRRs are in a moment. The Drosophila toll-1 is actually the best studied out of this particular group of receptors. And the hypothesis is that the MCC TLR activation occurs indirectly via endogenous TLR ligands. So leucine-rich repeats are used in lieu of the adaptive immune system, and will be an ongoing element of very ancient immune systems that will be repeated throughout this talk. This allows the binding of endogenous or other ligands, and then the TIRs, there'll be more detail on later. So prototypical TLRs, they're the tier domain that we just discussed, the leucine-rich repeats, and the transmembrane domain. Prototypical TLRs are not in porphyrians. Porphyria are sponges. They're basically living fossils that are simple multicellular organisms that lack true tissues or organs. But they do have the TLR NF-kappa B pathway intact, but it depends on which tissue that they're in that they're expressed. Prototypical TLRs are in Cnidaria, these are Cnidaria, and lack NEMO and independent I-kappa B homologs, but they do have a role in early development. Cnidaria are non-living jelly-like substances that are sandwiched between two layers of epithelium that are mostly 1 cell thick. So they're very interesting. They look pretty cool. Bilaterians, they're the larvae, and then the more organized adult Bilateria, which they all basically come down to being worms. So early ranging phyla predating the Bilaterians have prototypical TLRs, TLR-like proteins, and NF-kappa B pathway involvement. TLRs have 3 domains. They have the LRRs that bind up HAMPs, or endogenous ligands that we talked about before, transmembrane region, and then the intracellular toll IL-1 receptor, or TIR domain, which binds to the MyD88, MAL, TRAP, TRAM, and TRIF. As you probably have notice, the prototypical TLRs and TLRs are very similar in structure. My understanding their difference is actually which proteins that they bind to downstream, as noted here. So C. elegans actually lost most of their TLR to NF-kappa B signaling pathway components. The best studied in Drosophila as well as C. elegans are these TOL1, which is the prototypical MCC TLR that is this Drosophila TOL1. The naming convention is very confusing. And it's a tier-only protein. It's required for early development, promoting pathogen avoidance in the development and function of microbial metabolite sensing neurons. And then it does not signal to NF-kappa B. Mollusca express both SEC and MCC TLRs, and TLR-dependent expression of NF-kappa B pathway components as well. I probably lost most of you because at this point I was pretty lost as well. So I tried to go kind of evolutionary in order. So hence sometimes I feel like the definitions are a little bit unclear. This is basically just summarizing that in Drosophila there are 8 TLRs. They mainly detect fungi and bacteria. Humans have 10, and then mice actually have 13.
Allie, in this continuum of these, go back to that slide. Where do these things live? And I guess more importantly, what do they have to protect themselves from? I mean, we sort of know what we have to protect ourselves from.
Yeah.
You would think these things evolved because they had certain risks of what was going to kill them.
Right. I actually have more slides coming up.
Okay.
To summarize that. Where they live is usually actually in the water.
Yeah. Okay.
Yeah. But yeah, I have a good chart that actually talks about like selection pressures and criteria. So this is kind of this is along the lines of what you just asked, Len, but this is only for TLRs. The evolutionary pressure was largely in viral sensing, especially with TLR3 and TLR7. And then there's a high conservation of TLRs in vertebrates due to the need to maintain endogenous signaling networks. So this is kind of overwhelming, but this we know well from knowing our TLRs. But the function, there's some details here that we don't talk about as much, which is kind of interesting that this is what they highlighted. But the interspecies study, and most interesting is talking about the evolutionary pattern. What causes a strong purifying selection versus a high or low positive selection rate. Some like with the viral, it's very strong purifying or selection for both of them, but the rate of positive selection is actually low. Complement. We're finally to complement, guys. All right, so C3-like protein developed over 1 billion years ago in porphyria. The C3 is basically the integral defining protein that we base most of our studies about complement on, because C3 is not only shared but by multiple branches of the complement cascade, but also because it's because of its importance, it's most strongly conserved amongst all the different organisms. So C3-like protein with the homolog of factor B and MASPs may have formed a primitive alternative pathway to opsonize. There are conserved features of C3 that include chain structure and an internal thioester bond. Multiple gene copies of C3 and factor B in cnidarian species exist with different binding affinities. So what they think happened is that it throughout the millennia as evolution occurred, there were duplications in this region of the genes, so that this is how this occurred, and then it ended up being beneficial because these animals or these organisms didn't have they don't have adaptive immune responses. So having the ability to have different binding affinities to various ligands or antigens, I should say, is very, very helpful for these organisms. So in the terminal pathway, C5, 8, 9 were first seen in nurse shark, as well as equivalent components to C1, 2, and 4. And then complement receptors suggest that in sea squirt, they're suggested to exist in sea squirts. This is a phylogenetic chart or evolutionary chart of how the complements have kind of developed over time and what's added. There might be early alternate pathways, and then you start having antibodies present at the like the lamprey hagfish, but they're not true antibodies like we think about antibodies, and I have a few slides about that later. And then C3, and then the more classic complement cascade develops as we progress. So there's multiple theories on how all these elements of the immune system progress through history and through time. This is the most prominent and the most recent of all the proposed theories. It's first that the intracellular opsonic and cell activating cascade of single cell organisms occur or form. There's function on plasma membranes and secreted into intracellular space. Then with development of the circulatory system and hepatic synthesis of complement components, modern day complements developed, and now they're able to recognize foreign and altered self, opsonize and lyse membranes, as well as induce pro-inflammatory and survival response. The takeaway point from this particular figure is that they were looking at this short consensus repeats and how they evolve over time. And what's interesting is that these circles are basically areas that they're not really sure what order these branches kind of come off from. So even though we've been studying this system for decades now, probably just looking at the literature since at least the 60s and 70s, it's really interesting that they've grouped all areas, and you can't really see, they don't really know where the branch points are. Yes, this figure is from 1991, but the description of the literature that's a lot more current hasn't really discussed this in any detail at all. So moving on to cells, cellular innate immunity. So heterophils are homologs of mammalian neutrophils. Fish allow the discovery of novel inflammatory pathways, and neocytes, which are kind of like an IL-2 kind of cell, natural helper cells, innate helper cells, two cells, and multipotent progenitor type 2 cells are implicated in immunity in helminths and mammals. Neutralizing antibodies are what I was referring to earlier. Not natural antibodies. They're not adaptive. They just are made. They're largely IgM, and they're found in vertebrate plasma. I didn't know that these existed, so this is pretty cool. They basically opsonize what looks like non-self. Very limited repertoire, not adapted at all. It's part of the innate immune system. And you can see that it's expressed in multiple different kinds of organisms in the phylum and phylogeny. And they basically help neutralize pathogens, helminths. They have provide some resistance against influenza, the bind oxidative proteins, and they allow for immune surveillance against tumor-associated or tumor-specific antigen. Phagocytic B cells, super cool. I'm a B cell biologist and I didn't even know that these existed. They're able to produce IgG and IgA following classical adaptive immune responses. They make natural antibodies. They phagocytose and activate degradative pathways that result in phagolysosome fusion and killing of internalized bacteria, much like we see in our macrophages and neutrophils. It's been identified in fish, reptiles, and mammals. And this is where they talk about mammals. There's B1 cells that are of questionable existence, but seem to have active responsibilities in the peritoneal and pleural cavities. And then from bipotent B macrophage precursors in the fetal liver and adult bone marrow, this supports that B cells and macrophages have a common phagocytic origin. Intraepithelial T cells recognize and destroy pathogens. They're clearly better characterized in mammals. They haven't really looked at it that well in other organisms. Most of these are gamma deltas, and they're most frequently found in the epithelium of the intestine. They're seen in amphibians, fish, birds, and mammals. And humans have both a V delta 1 and V delta 2 gamma delta, where delta 1 resides mostly in the epithelial mucosa, and delta 2 are abundant in the lymphatic system and peripheral blood. So we're going to move now to the adaptive immunity. I'm running a little short in time, so I apologize. I'm going to talk a little bit fast and skip over some details. Vertebrates, so they basically divide everything, all the organisms into jawless versus jawed vertebrates. Jawless, there's about 100 species of hagfish and lamprey. And then there's over 10,000, there's tens of thousands of species, I should say, of jawed vertebrates, including mammals. And then the best part about vertebrates is that they have lymphocytes. So this is a fun depiction of what a lamprey might look like. It was larger than human. This is from June. I was talking to Karen Chen yesterday about my talk, and we were talking about worms, and this came up, and I figured it'd be fun to have a GIF of a worm. So lampreys are the best studied out of all the organisms, as lampreys are at that bisection of jawed and jawless, and they're direct descendants of primitive vertebrates of greater than 400 million years ago. So they're kind of an animal that allows us to go back in time, despite the fact that they're still alive today. They're kind of interesting looking with because they're jawless. They have this kind of scary looking face. They have some adaptive immunity, but no antibodies, no T cell receptors, MHC or RAG. What they do have are these elements that are called, these genes called variable lymphocyte receptors, or VLRs, and they are comprised largely of leucine rich repeats. VLR A, B, and C are incomplete VLR germline genes, but VLR A and C they code for receptors that form two different T-like lymphocyte lineages on cell membranes, and VLR B are expressed in B-like lymphocytes. There are two forms of velar B. One is anchored to the cell membrane by a GPI, or it's secreted as an antibody molecule by mature plasmacytes. And it's thought to be that they form multimers when they are secreted. Cytosine deaminase 1 or CDA1 is involved in VLRA assembly, and CDA2 is involved in VLRB assembly. Again, VLRA1 is more of a T cell-like receptor versus VLRB is more of a B cell-like receptor or component. And then on B-like lymphocytes, these elements bind bacteria or erythrocyte antigens. They cause activation, proliferation, and differentiation into plasmacytes that secrete multimeric VLRB antibodies specific for protein or carbohydrate epitopes. So what's been really cool is that C. lampreys also have basically they're like thymuses. They're in the gills of these animals, and they've been seen for the thymic transcription factor FOXN1, as can be seen here with a good demonstration of existence. FOXN1 interacts with their target gene DLLB to cause activation or transcription, and then in the gills, this is just a demonstration that it's in the gill filaments largely where these cells are expressed. FOXN1 likely regulate chemokine expression in these thymuses. DLL4, which is a homolog of DLLB, is it's a Notch1 ligand and directs progenitors T cell lineage. The positioning of the thymopoietic tissues in the pharyngeal region is conserved, but the location and organization varies. Right, we just talked about that it's in the thymoids in the lamprey, and then in mammals in the thoracic thymus, but clearly up also in the neck area. This is a illustration of just at what time point usually about where these different organ structures become evident in animals, and the lymph node is the last to develop. So lymphocytes, despite my interest in it, there's very little known about prevertebrate lymphocytes and its role it really plays in phylogeny. The telos and shark data indicate that there's a spontaneous natural killer cell like cytotoxicity against parasites, but they don't really know how it kills. There's these SPY1, SPY-B, and SPY-C lymphocyte transcription factors that interact with a DNA binding domain called ETS, and that turns on genes of import in lymphocyte development. And then the prevailing hypothesis of vertebrate immune and MHC immune system function is protection from parasites with distinct genomes. So this is just a demonstration of how antibody or antibody-like development has occurred in invertebrates versus prevertebrates. In vertebrates, there's RAG-mediated rearrangement, and then there's a rearranged gene from that, and then they're able to progress. If it's dependent on the leucine-rich repeats, those are RAG-independent rearrangements. There are RAG elements that are inserted for adaptive response, and then they form VLRs instead of IgGs or Igs. I apologize. So T cells and histocompatibility complexes. I group these together because we just know so little about both. MHCs and other histocompatibility complexes. So if you don't have an MHC, the animal is not going to have RAG or TCR. Only jawed vertebrates have MHC. So as I demonstrated earlier, it comes much later in development of the immune system. They were first found in cartilaginous fish, and the MHC loci vertebrates are largely conserved in metazoans. T cells, alpha and beta, are largely conserved, and the gamma delta innate functions with large variability with genetic layout and functions exist. TCRs were first found in cartilaginous fish, which is in line with what we just discovered, where MHCs were first discovered there. So immunoglobulins first occurred with vertebrates. They were identified in bony fish, but absent in lampreys. And then IgM was secreted is secreted as a tetramer in bony fish. And overall is evolutionarily conservative, as we previously discussed. Nathosomes use the immunoglobulin superfamily members and are used in TCR, BCR, MHC, invariant NK receptors, phagocytic FC receptors, cytokine receptors, cell adhesion molecules, as well as TMB interacting cell surface molecules. As previously discussed, nathosomes are all the way, in this area. Sorry, this area. They're RAG-mediated rearrangements, and nathosome lymphocytes and molecular machinery are found in lampreys and hagfish, but agnathans actually use LR insertions to generate viability. Adaptive immunoglobulins require RAG in order to form. RAG 1 and 2 is actually fascinating. They actually are very close in proximity. They actually don't have introns. They're present in sharks, but not in protochordates. So this implies that actually we all inherited this by a viral mediated retrotransposon. There are also other various elements about the adaptive immune system that is discussed, but nothing more than really what's been summarized here. There's an immunoglobulin called IgY that's been evolved to become IgG and IgE. Immunoglobulin new antigen receptor, or IGNAR, has a V region that's conserved in some TCRs. And then the light chains gamma, delta, I apologize, lambda and kappa emerged from a common ancestor to all living vertebrates. There's a third kind of light chain that's called the sigma, and this was first seen in all cold, or is seen, I should say, in all cold-blooded nathosomes. The heavy chain class switch first evolved in amphibians with mammalian-like Ig heavy chain loci. Bony fish IgM and IgD heavy chains is generated by alternative splicing. IgT is another isosite that is generated by alternative rearrangement, and cartilaginous fish do not class switch between IgM and specific other antibody heavy chain classes that are specific to them. All sound very kind of disjointed, and I don't have a good story here, and that's true. It's because there's just so little that's known about antibodies in the immune system development, especially with regards to phylogeny.
Ellie, does preparing this talk help explain IgD? Why we have is it just a carryover of no biologic function? I've always wondered why we have IgD. We know almost nothing about it, and it doesn't seem to do anything.
No, because it's lost in most species. And
It's in my, yeah, but what does it do for us?
no one knows.
So, I mean, you get the sense that it's just a historical carryover that we still make it, but it's functionless in humans.
That's my understanding from,
Okay.
yeah. People don't, I was lucky that this figure even had IgD. Nobody talks about it. They just pretend that it's not there. And this is the coolest part. So a few years ago, this gentleman, Dr. Ono, proposed that, actually it wasn't a few years ago, I want to say it's probably in the 80s, 90s, proposed that the vertebrate genome underwent 1 to 2 rounds of whole genome duplication at the stage of fish or amphibians through a tetraploidization process. And it was only actually relatively recently proven that this is actually real. They've demonstrated that, or proven that there's definitely 2 rounds of WGD that occurs after the emergence of urochordates and before the radiation of jaw vertebrates. But there's a potential third here with the bony fish as well. And it's with this kind of system that these, like, we've been able to develop a good, robust immune system because of this, and that we have various abilities to adapt to our surrounding environment based off of all of this. So
So are you saying at some point in time, living organisms just doubled their whole genome?
yeah,
Carried 2 of everything, twice as
Yeah.
much genetic material?
mm-hmm. Or
Why'd that happen?
other than selection pressure, it's not really, that's basically it's a very hand wavy explanation. They don't seem to really know why. They just that's
Maybe they did.
how to-
Maybe they did it to make the board exam twice as hard.
Yes.
Yeah.
They did that's their goal. So now, yeah, we have so many more things to study for. At least we don't have to know lampreys, guys. They are really cool animals. But yeah, the word of the day is tiger. Thanks so much for listening. I'm sorry about the technical difficulties from the beginning. And if you guys want my bibliography, here it is. But I'll put this back on the word of the day. That's the key thing for this morning.
So I'm sure this would be a hard thing to answer, but if very simple early organisms that had no adaptive immune system, had a very primitive immune protection, why do they still exist? You would think if they couldn't protect themselves very well, they would die off.
So it depends on what kind of organism that they're protecting themselves against. So if the earlier on the organisms, they were protecting themselves against parasites, helminths, and then over time, then that also included bacteria, and then eventually viruses. So my understanding is that's really what drove the evolution, and that's why lampreys still exist, even though they phylogenetically branched off so early, and they're still able to protect themselves. The leucine-rich repeats do a pretty good job protecting them.
Well, that raises the parallel question. Do we know when in the history of the Earth parasites and pathogens developed? Do we know when viruses developed and bacteria, gram-negative, gram-positive, all the things that we have to protect ourselves against? Have people done parallel studies to try and answer that question?
Yeah, I didn't have time to go that deeply into that, but yeah, there was a lot of suggestion that they've just in the discussion of of all these details, there was a lot that implied that there was had been a lot of investigation into all of that.
Oh, well, good. That's a good talk for next year. That makes it one less talk I have to invent for next year. All right, we've still got a pretty large audience listening to you, I think. There are over 30 people. We have other queries
People
out
stuck with
there.
me.
Yeah. Anybody else want to chime in? Well, if not, what does the word of the day have to do with this talk?
I don't know.
Oh, you didn't make it up?
No. Meryl sent it to me. Maybe I should have asked for phylogeny or something like that.
Okay.
Or lamprey.
All right. Any other comments before we sign off?
Oh, there's a chat.